We're down to a sort of a hardcore here, but I wanted to have a chance to do a check-in at the end. And I hope it was productive. People are welcome to keep using the Zoom to discuss afterwards. But I thought officially we were going to have a little wrap up at 5 o'clock. So I wanted to make sure everybody had the chance to do that. Do you want to, shall we just go through the usual order? So tissue engineering people, were you able to get, how did it go after I talked to you?
So for me, well, so I think I have now a simulation of this, and they do align when we pull on one of the compartments. I can maybe share it to see it. I mean, it's
Oops.
not, I think, for the time, it's okay. So maybe so now I'm pulling on this green compartment, and sometimes it works better than in other times because it also randomly chooses the compartments. And when it doesn't choose compartments in this area so much, then we don't see the effect as much, or it takes longer for the simulation. And then when there are other stress fibers that are pulling, then there's also tension in other directions. So it's not always as good as I would for it to be, but I think we can see some sort of an effect.
Well, one way maybe
to
work on that is to have the lifetime not go to infinity. In other words, even if there's a strong stress, there's still some probability of having it decay.
Yes. Yeah.
So you could change the bottom part of your sigmoid for that.
Mm-hmm. Yeah.
But that's aligning pretty well. That's aligning pretty well.
Yeah.
I mean, if you look at real stress fibers, there always are ones that are in the wrong direction.
Exactly. Yeah. No, so I'm very happy with what I came up with, and I wanted to say thanks because I did not expect to get this far on this hackathon, and I was really very positively surprised. And thank you very much.
Well, thank you for being patient, and if there are things we can do to help you make this something that's useful to you, please do stay in touch with us.
I will.
I think it's a great problem.
Okay.
I don't know if Kai Chun, was she able to get her pillars to work at all? Do you know? She's not on the call now. Oh, there she is. Kai Chun, were you able to make any progress?
I just put. Can you see my screen?
Yes, it's.
Yeah.
Oh, that looks good.
Yeah, that is what I want.
That's pretty amazing. You were able to get that all done since the last time we talked?
But this is the basic step of my model. In a following work, I will add some sectors that I am interested in experiments.
Well, that's great. I'm pleased that that worked.
Oh.
And now you could add the stress fiber model that Rebecca is doing and have a really nice, great. Well, I'm impressed. And it runs not too slowly, so that's great.
Yeah, the parameters in a current model is not it's a final version. I will adjust parameters more carefully based on the experimental results.
That's great. Yeah. I mean, I have to say these are both of the things that we've seen here are things we've wanted to see somebody do for years. And so I'm really pleased that you've done them, because we've always wanted to be able to do these. We've always known you could do these things, but nobody actually took the trouble to do them. And so it'd be really nice to be able to show that you can do this kind of modeling.
I really appreciate your help, you and Bobby and help.
Okay.
And the
Does
simulation
anybody?
speed is very fast. So I'm really excited.
One thing I'd like to point out to the group is that, and this is something we kind of learned over the course of the weekend, beginners and intermediate modelers, we often try to develop a model that looks real, that looks like reality. But we made a lot of progress when it was pointed out in different instances that we need to do some things that look somewhat unrealistic, but provide more realistic behaviors. In Kai-Chun's model, which you can see there, the filaments are floating in air and they're held up by these links, or held in place by these links. That looks much less realistic than, it behaves much more realistic, but looks much less realistic than the original version, which was a 3D vertical stack. And that didn't act right at all. So I think, reminding, that's a abstraction process that we've hopefully we've improved upon over the course of the weekend.
You're muted.
If people are willing to share their intermediate code, that would be really helpful. So if you can save a zipped version of this to that shared directory, then we can have that. We can download it and work with you on it. That would be very helpful. I mean, this one you've done in Kai-Chun is particularly nice to me because there was this work that was done by Merck's student, or Urs. And they used a full solids modeling package, a very elaborate solids modeling package. And I think you could actually do almost everything they did with this. The one thing you might add would be crossbars between the pillars. So your pillars are freestanding because your pillars really are freestanding. But you could add links between the elements, and then that would make it into a solid material. That would be like a cell crawling on a gel. And I think that would be interesting too. So I think what you've got there is actually a very good basis for a more detailed model, and especially combining it with that stress fiber model, one could do quite a bit. So that's great. Okay. Well, I hope that was useful. I know it took a lot of your time, and it's very different from the work you've been doing on signal transduction. So I appreciate your taking the time to work on the CompuCell side. All right.
These were the cellular model and the signal pathway model. Both of them, I am really interested in both of them. I hope I can combine two kind of model together to investigate underlying mechanism detailing. And then some of my students can do some experiments. So I hope I can combine the experimental results and the simulation result get together.
Right.
Oh, I think it will be very good.
But I'd like, even if it's very abstract, and frankly, a methods paper on both of these things would be very nice. So I'd be happy to talk to you. You're local at the moment, so we could talk more easily. But that's great. I appreciate that. Great problem. Anybody else want to say anything about from the tissue engineering or group, or shall we move on to the thymus people? How is it going?
Hi. Oops. Yeah, yes. So it's weird. I cannot see my video. Can you see me?
I can see you.
Yeah, okay. Yes, so today, the first part I work with Anurag, and we have, yes, looked a bit more at all these ODE system we have found over the last few days, and it's quite interesting to see that I think it would be almost possible to write one big system that when you take the limit to some parameters, you can go basically to all of them. Like just they are different, but you could just take one parameter to 0, and that you would just jump from one model to the other. So we have written a model in Tellurium, and we just need to do some parameter fitting. There's not so much data, so I need to email few groups that show data in that, but it's a bit complicated trying to extract data from a graphic, and they don't provide the table. So yes, so that will, after that, I guess we can do parameter fitting, and that would be useful to implement that in CC3D. Otherwise, yeah, I had like 30 minutes after he left to just go back to the CC3D and just play a bit with it. So I can share that. He's doing some funny thing. So the blue cell are basically one type of progenitor T cell that are supposed to move up, and when they reach this part here, they change type, and they are supposed to go down. But obviously, everything is crowded, so they just don't move anymore in the good direction. So, I mean, I was not expecting this to work. So because I haven't thought about the real parameters, but we need to make this behavior by letting some more space between the cells, and probably think about how they can move in both opposite direction at the same time. Yes, and use different types of fields for different type of secretion in function of the different parts. That would be the next step. Yeah, we also built a list of the most important thing we want to do in the next few weeks, or for those who are interested to still working on it. I think there are like a few people that still want to work on that, so that's nice. I cannot hear you. Sorry.
I said, I hope it was productive. I know
Yeah.
it's a long 3 days, but I hope it was, you were able to pull together. It sounds like you've got a good agenda for going forward with this.
Yeah, yes.
And again, we'll do our best to support you on it. I think, again, everyone had a nice problem. They were all good problems.
Yes.
Every now and
Yeah,
then
somebody
comes to a workshop and they want to do something which you just can't do with CompuCell. Somebody wants to do molecular dynamics model.
Yeah.
And you say, well, it's a nice problem, but it's not the right software. But I think pretty much everybody's problems this time were appropriate, and I think that's great. So, is there anything else you wanted to say before
Also, thank you for the organization and all the stuff we've been able to learn these 2 weeks, and yeah, that was great. So yeah, and I'll probably email a few of you in the next couple of months with a lot of questions, I guess. Yeah.
Well, we're happy to set up Zoom sessions with people when they need them, and
yes,
keep working on it. Okay.
Thanks.
Okay, great. Thank you. Let's see. Is there anybody left from vascular tumors? I don't think so. Bacterial motility. It's very late for you, so we want to let you get some sleep.
Don't worry, I'm used to it now. So we have implemented the extra links to try and implement more of the bending, and we've been looking into essentially what we can really answer with what we have implemented so far, because we've reached the point where there's lots of things we could now add to it, but there's so much that we could already answer with what we have. So comparing it to one of the papers we've kind of based the model off, varying how the active force of each of the cells relates to the length of the filament and the bending, and how that all results in the formation of different shapes of the filament spiraling or staying on a straight path, that kind of thing. That's all stuff that we could look into with it so far. And coming back to our sort of original question of how much does each cell contribute to the collective motion of the filament, I think we've been able to at least answer that a little bit with what we have so far, in seeing that the filament motion definitely needs to be driven in some way by the lead cell to prevent any of that bundling that we've been seeing, even no matter what the bending is implemented or anything like that. There's loads of places for us to go forward with this. We had a look at implementing some secretion earlier, but we haven't. I think that's on Hariana's version of the model. But implementing some secretion, looking at the phototaxis, looking at the variation of the different parameters to see if we can rep some of the shapes and paths of motion that we're seeing in the data, and potentially working on a compartmentalized model to compare this to so we can kind of compare how the orientation of the cells in the chain matters to whether there's a polarity to the bond, and whether the reversal of that polarity is what allows the filaments to switch the direction of their motion, that kind of thing. So yeah, lots of progress and lots of places to go forward. So.
Great. Well, I'm glad you got. I mean, that looks like a reasonable place to start. I know it's frustrating getting these things going. The bootstrapping is always frustrating, but once you have something that works, developing it is easier. And so definitely. I appreciate that. So who is Evan? Did you have, it's okay, before we stopped, is there anything else you wanted to talk about now?
I guess just thank you very much for the opportunity to take part in this, especially as an undergraduate, and this being my first foray into any kind of research. This has been a really brilliant learning opportunity for me. So thank you very much to the organizers for everything, because it's been really great.
Well, I should say I'm delighted. I would never have known you were an undergraduate versus anything else. And I think you mentioned it, but we've had literally everything in these courses over the years. We've literally had everything from high school students to emeritus professors. And we've had everything from surgeons to pure mathematicians. And so one of the things that I like about this course is that nobody comes in knowing everything. And so there's a certain level of quality. You have to be willing to try things and not worry about breaking them. But we're all ignorant. The biology is very complicated. The computing is a pain. And so I think one of the things I like about these courses is that people could collaborate as equals in these things. That sounds, I'm sorry, that sounds a little condescending. I don't mean to be at all.
Not at all.
On the contrary, but I'm delighted that it was productive for you. I hope we can keep going forward with this.
Absolutely.
And again, I think everything so far, all of these things are right. Could be written up. It's even the basic things you've got could be papers about methodologies and ideas. And so I hope people are able to take these ideas and turn them into things that you could publish. One thing that we didn't talk about, which Giuliano has worked on, is people have moved rapidly away from using NanoHub, I see, which is fine. But one of the things you could do with NanoHub is, if you've written a simulation, you could put the simulation up. Remember, we ran that COVID model as a freestanding simulation. And so you could put your simulation up on NanoHub, and then somebody can run it without having to download it and install anything. and it gets to DOI, so it actually counts as a publication. And so if people have sort of versions of their programs as they develop that they want to release that way, Giuliano, it's not the easiest thing in the world to put things. NanoHub is a bit of a pain to get things installed. But Giuliano has done some nice work on making it as easy as possible. And he can walk you through the process if you decide you want to take your model and make it something where people can run it on the web. He can help people with that. So just to make people aware of that. We use it primarily for educational purposes. With those NanoHub things, we use primarily for teaching. But you could certainly run research code on it too. So that's something to think about as a distribution mechanism, because, well, there's something to do. That's great. Well, thank you. Okay. Sorry. Let's quick before. Who else? Was there anybody else? Was there anybody from the lateral line group that's left? Evan, did you want to say something?
Hi, sure. I got a little bit stuck. It seems like I had really good luck all weekend, and then I got to the very end and I hit a road bump. I'm working on a parameter scan, and I added a JSON file, and I get the pop-up when I go to run it in the player. I'm trying to share my screen, but it's not working. I don't know why. Oh, there we go. Okay. So yeah, okay. So I've added the JSON file, which I guess for me opens as a notebook. So I'm just scanning over 5 values, and I go to CompuCell. Oh, sorry, I should start with Twedit. Where did it go? Yeah, and now I open and I get this pop-up and it's great. And I display the command line and I just run. I think this is right. Maybe I should use GUI. And I just sit there. So I'm not totally sure what's going on, because when I run the demo works, but this doesn't. So I'm a little.
So I'm sorry about that. I am not afraid to say I don't know. And the answer, what I will say is I don't know, because the version CompuCell 4 has a different parameter scan utility from the one that was in CompuCell 3. And I have never used the parameter scans in CompuCell 4. And so I would ask you to zip it and send it to Maciek and have him look at it for you, because I literally have never used it.
Okay, that sounds good.
And I mean, TJ and Josh do a lot of parameter scans, but they typically use their own Python scripts to do it. They don't use the built-in.
Okay.
And so I can't help you with that one.
That's okay. That's fine with me.
Is the code, I mean, was the simulation inside that working at all? I mean, in other words, besides the parameter scan, if you're scanning a parameter, was there a model that was working that you wanted to scan the parameter for? Okay, so at least we made some progress there. That's what I
yes.
And I apologize for that problem with the parameter scan, but I can't, that one.
That's okay.
I can't help that. But if we can help you more on the modeling part of it, I'd like to do that.
Sure. Yeah. And thank you as well. This has been fantastic. I got way further along than I thought I would in 3 days, for sure.
Well, thank you. I know it's a long, I mean, people who went through the whole thing, it's 2 weeks of your life. So it's a lot of time. So the people who've hung out to the end, we very much appreciate. Josh, did you want to talk a little bit about where you are with your model?
Sure. I have it snapshotted so it can be brief. So take the screen share here. So the progress on the model is, I had hit some an interesting point thinking about diffusion in the skin, and it really building the model really forced me to examine like assumptions and look for approximate values for diffusion of some of the proteins in the dermis. And in working with the folks today, it seems that the diffusion of the proteins that are deposited by the insect during the bite diffuse at a much faster time scale than the recruitment of immune cells to the site, let alone infection with the leishmania, which requires the leishmania to get phagocytosed. So we got as far in the model as being able to initialize the epithelial layer and put the little bolus of leishmania parasites as well as a bolus of saliva, initializing that one time step before this was like right over it. And this is the first MCS step. So this is about 1 minute showing the diffusion of the saliva proteins that were deposited right there. So, you know, it's a matter of thinking about we talked about what's the best diffusion solver for it, which solver offers the flexibility to illustrate, to model the kind of chain of events in immune signaling and recruitment, because the saliva, now you have to, what I'm currently working on is with Dr. Glazier is detecting the amount of saliva in the epithelial cells that will trigger those epithelial cells to start secreting chemokine, and those are going to recruit neutrophils, which are in turn going to recruit macrophages. And if they get the macrophages that get recruited to this site, the ones that eat the leishmania or the poor hapless souls that get infected. So the next steps of the model that need to be developed are pretty well, I mean, in terms of concept, we kind of know where we need to go next. So that's good, and I'm curious to see the behavior of the system when all that is made. What Dr. Glazier and I were talking about just before this wrap up is it will be cool to simulate this interesting dynamic of the neutrophils eating the leishmania, and then some of those neutrophils undergo apoptosis, and those get eaten by the macrophages, and that's actually a really important way by which the macrophages get infected with this parasite. And then when those macrophages die, they break open and release more of these little critters, which again don't really move. They just kind of wait for more inflammatory cells to come to it. So it'll be interesting to see how all this works, because I kind of assumed this would all be via, you know, chemokine and chemotaxis gradients. But given the rate of diffusion, I don't really know how strong those gradients are, because I don't know that the leishmania aren't secreting anything to our knowledge. Well, okay, there might be some new data. I won't get to that. It is not really thought that they're secreting anything that creates a gradient to attract things to them right now. So yeah, I'm the same way with the rest of my colleagues. I'm really thankful to all of the organizers of this. This has been real. I've been wanting to get into modeling for some time. Again, I have some of those kind of grandiose, Dr. Glazier, what you talked about at the first day, ab initio modeling dreams of kind of incorporating this and using this for vaccine design and trying to, you know, I'm bothered by, and you see this in the discussion about COVID-19, of, you know, everyone's focused on neutralizing antibodies and do you have enough neutralizing antibodies? There's all of this focus on like a single parameter, which is of course incredibly important, but I think the problem that we have in the field as clinicians and immunologists is that we conflate that one parameter with immune protection. And protection is an emergent property of the system that we really don't understand. I mean, just speaking, to be on the soapbox a little bit, we don't really know how vaccines work, period. I can show you like RNA-seq data and like signatures of people who are protected, but I don't know that I would like to know more about why that is protection. So that's the grandiose goal. Right now, I just need to get this to work. So I look forward to working with more members of the CompuCell team in the coming days on this model. This has been really educational and fun. Thank you.
Thank you. And I'm happy after we've had our recap, I'm happy to go back and break out and talk a little more. People are welcome to use the Zoom session for a little longer if you want. We don't have to end it at any particular time if people want to keep using it. There are a couple of people we haven't heard from, Joel, Haryana, and Sandeep. Did you want to have some last comments before we wrap up?
Yeah. Actually, go ahead, Sandeep. Sorry, this is Sandeep. Well, I had two ideas to begin with, but I couldn't quite implement it. But I'm looking forward for the next hackathon. If that happens, that would be great. But otherwise, the initial part of it, the OD modeling, that's something which I'm pretty much familiar with, and that was pretty good. And also the second day was good, and after that I lost track. But that was a good amount of learning, I would say. And other than that, so I did not do much in the group, but I did understand what they were trying to do, which was again good. Yeah, but I would like to do something which I have two ideas, which one of which I discussed with Bobby, and I would like to work on it in the future.
Well, again, we're here to try to help. So
thank you for organizing this. It has been a great workshop. Yeah.
Well, thank you for sticking it out. I appreciate it.
Yep.
Okay. Anybody else before we break?
Yeah, I just want to say thank you for organizing this fantastic conference. I was here for the past week, and through the hackathon, it was a lot of fun, a lot of learning. And I thought you guys did a great job with all these exercises and running through the code. And I think I especially like the part where you're able to integrate the ODE into CompuCell 3D. That was not something I was familiar with and will definitely make use of in the future. And I just learned a lot of other things, especially this hackathon too. I think working on the model with Katie and yeah, building a model from scratch, defining the problem was really, really great. And maybe next time, I think I would like to, there's been a problem I've been thinking about also that building in CompuCell 3D. So maybe with your guys' help on another hackathon, we could build that and yeah.
You don't have to wait for a hackathon. We can try to help you out. We can meet with you. I mean, the only, of course, the world, life happens. And so the problem is outside of these hackathons where we carve out really 3 days to be available.
Mm-hmm.
Now we could give you an hour or two here and there, but that could work too also if you do it every week or every 2 weeks for a while. So we're definitely happy to brainstorm ideas and help get things going. So
all right.
you don't have to wait for the next hackathon if you don't want.
Okay. And the best way to reach you guys just through email then?
People are grinning because I always say if you email me and you don't hear back within 48 hours, call me. Okay. Because I get so many emails, I lose them. On the other hand, TJ and Maciek and Andy are very good about answering email. I don't know quite how they do it. And so I would recommend CCing a couple of us so that we know. Jim Sluka is also very good at answering email. So I would recommend making sure that there's more than one person on your email list.
Okay.
Because I tend to get a bit overwhelmed. But usually if you don't get an answer, it's not because I'm ignoring you, it's because I didn't see it. Because it just gets buried. The email gets buried.
I understand that, yeah.
Right. But I really like working on these projects with people.
Okay, it's good to know.
And I'm sorry I didn't have more time. I tried to circulate through all the groups, but I didn't get to talk to everybody. And so
that's all right.
I hope everybody got some help. Okay, yeah. Great. Joel, I guess you're the last
Yeah.
person on the.
I think I have to say something because Romain expect me to thank you all the people involved in putting this together and make this possible. We basically got everything that we were aiming for. As I can share my screen here. So this was the ultimate goal that he has before doing the models. And right now we got everything that we put it into effort to do. So it looks very good. You just have to adjust now parameters. You can see by the dots that it's a little bit off of the ODE, but it's going there, it's getting there. Now it's just adjusting some parameters. So it's been very, very educational, especially for me, because I don't have any knowledge on modeling. My background is in computer science, and this has been an awesome experience.
Oh, wow, that really does look impressive. I mean, it's tracking very well. So
Yeah.
that's good.
If we take
Did Josh help you with that?
Yeah,
That
Josh,
looks like one of Josh's simulations.
Yeah, Josh helped us at the beginning specifically to make how to make a cell, generate a cell in the middle, and organize stuff. Yeah, and Gilberto has been very useful for us in terms of how to select a specific random cells. He was very good at helping us with that. Yeah, it works very well, and it's doing exactly what we were expecting on every point. I think he will just work now on defining better the parameters and make it follows the ODE as it's supposed to do.
Okay, well, thank you, everyone. I appreciate your sticking it out. It started with 70 people and we're down to what, 10? So you're really, you're troopers. You're welcome. As I say, I'll leave the Zoom on for another hour, an hour and a half. There's no reason we have to turn it off. It can run as long as it likes. And so people are welcome to discuss here if they want. And with that, I would say I look forward to hearing from all of you. If you're willing to share intermediate code, that would be great. So we can have that to look at and work with. And we'll look forward to working with you as we develop these projects. And again, I think Bobby will be sending some questionnaires about organization and improvements. Everything was a bit of an experiment this time. It's the first time we did it virtually, focus on COVID. Ironically, given how many developmental people there are, a switch to COVID may not have been so well chosen as our example, but I hope it was interesting anyway. And so we'll be hearing from us all in the next few days, and we'll be working with you as we go forward. So with that, I'll let people just say thank you again. Thank you, Andy, Bobby, Javier, and all the other people who were helping out. Yi was an unexpected one. I mean, Yi just joined and helped out because she's nice. She was my second PhD student a long time ago. Dominic Lynch, who helped out for a couple of days, he was a student in the CompuCell course last year. So it was very generous of him to come and help out, having taken the course last year. So, and Bobby, again, I want to thank him very much. Andy, also for helping out, and Javier, who are not CompuCell people, for doing so much. So with that, I'll let people, you're free to use the breakout rooms or to go have dinner and get some rest after this long haul. Josh, we could talk a little bit in the breakout room again if you like. I'll join you there in a second. So with that, our official program is over, and I look forward to working with you in the future. Thank you.
Thank you.